Okay, let's unpack this. For anyone following the cutting edge of oncology, CRRT cell therapy has been, one of the most life-saving, just incredible breakthroughs in decades.
It really has.
But let's be honest, it's also been incredibly complex, slow, and just astronomically expensive.
That's the crux of the issue, right? Autologous CAR T-cell, that means cells sourced and engineered from the patient themselves. It gave us these durable, even curative remissions for tough blood cancers.
We're talking relapsed refractory non-Hodgkin lymphoma, multiple myeloma.
Exactly. But we just couldn't scale it. The wait time for manufacturing, that prohibitive price tag, and the sheer logistical complexity, accessibility has been a massive barrier.
So the mission of this deep dive is to shortcut you right to the therapeutic frontiers where researchers are, actively demolishing those barriers. We've pulled sources detailing these lightning-fast advancements with a heavy focus on innovation coming out of China.
And we're tracking 3 parallel, really revolutionary efforts. First, making the therapy scalable and fast. That's universal CAR-CAT or UCAR-T, the off-the-shelf approach.
Off the shelf, I like that.
Second, making it more potent and durable. So using dual or tandem CAR-T products, TANCARs, to overcome the problem of tumor resistance.
And finally, and this is arguably the most critical piece for real-world adoption, we're looking at standardizing the management of those potentially fatal side effects. The dreaded cytokine release syndrome, neurotoxicity.
Yes. If we can't manage the side effects, you can't treat the masses. It's as simple as that.
Precisely. So let's dive into that first challenge, accessibility and speed.
Okay.
Current autologous therapy is personalized medicine at its most extreme. You harvest T cells, you ship them, you edit them, you grow them for weeks, then ship them back.
And that waiting period is clinically dangerous for these patients. And if the harvest fails, you're back to square one.
So the whole industry is looking at UCAR-T.
It has to. This uses allogeneic cells, so cells from a healthy non-patient donor. You manufacture them in huge batches, store them, and they're theoretically ready to go, ready to infuse the moment a patient needs them.
Now, this is where the real tech challenge comes in, right? When you introduce foreign cells, you have 2 major rejection problems you have to solve with gene editing. The first is GVHD,
Graft versus host disease.
the donor cells attacking the patient.
Right. And the second problem, which is often overlooked, is host versus graft rejection, or HVGR. This is where the patient's own immune system sees the donor cells as foreign and just wipes them out before they can work.
Okay, so if the T cells are the guided missile, GVHD is the missile attacking its own base, and HVGR is the base shooting down its own incoming missile. We need to disable both.
That's a great analogy. And to disable GVHD, we rely on precision gene editing, usually CRISPR, to essentially blind the donor T cells. We knock out the gene for the T cell receptor alpha constant, the TREX gene.
No track, no vision. The cell can't see the patient's tissues to attack them.
Exactly. But what about HVGR? Let's look at how innovators are tackling the patient's rejection of the therapy. We saw a powerful example from Northhang Bio, their CTA-101 product.
What makes CTA-101 so significant?
Well, it was the first domestic UCAR2 innovative drug approved in China based on CRISPR editing. And they have this fascinating double knockout strategy to make sure the cells persist.
So they handled GVHD with the trait knockout. What was the second knockout for HVGR?
They also knocked out the CD52 gene. This is really key. Knocking out CD52 lets clinicians give an anti-CD52 antibody as part of the pre-treatment.
And that antibody would normally kill the CAR T cells too.
Right, but because the CD52 target has been edited out, the antibody suppresses the patient's host immune system, preventing that HVGR response, but it leaves the CTI-101 cells totally untouched.
So they made the therapy invisible to the drug that's suppressing the host defenses. That is a highly sophisticated strategy to give the CAR T cells time to work.
And the early results seem to validate it. In a trial of 6 heavily pre-treated adult B-ALL patients, a really tough population, they reported an 83.3% complete response rate.
Wow.
That's a huge data point. It proves an off-the-shelf product can navigate that GVHD-HVGR tightrope in the real world.
So that's one path, using really clever gene editing. But we saw another path, focused more on massive industrial scale production from Boya Bio.
Right, Boya Bio is all about their Tiucell platform. Their product, BRL-301, aims for immune evasion without needing complex HLA typing, which is another time and cost barrier.
Okay, let's pause on the scale here, because this is where that cost barrier really starts to crumble. What kind of numbers are we talking about?
This is the industrialization of cell therapy. They achieved a production scale of over 200 patients per single batch.
200 patients. from one batch.
Think about that. The cost savings when you go from 200 separate bespoke manufacturing runs to one single optimized run. It completely changes the game on availability and cost.
That raises a critical operational question, though. I mean, manufacturing 200 unique patient doses, well, not unique, but 200 doses at once, that sounds like a massive quality control challenge.
That's a great probing question. And the key advantage is that since it's allogeneic, the source material is consistent. It's from healthy donors. So you have highly standardized input material and a predictable manufacturing curve.
Unlike autologous, where the starting T cells from the patient can be all over the map depending on their health.
Exactly. Standardization is just so much simpler with a standardized donor input. The QC is built into the allogeneic model.
That's a massive insight. Okay, before we leave speed and scale, we have to touch on another innovation, just the sheer pursuit of speed itself.
Absolutely. We have to mention Gracell's FastCAR platform. Now, this was for an autologous product, GC012F, but they demonstrated next-day manufacturing from weeks to days. It proves that speed is a global priority, not just something you get with UCAR-T.
So we've covered the first major challenge, cost and time, solved by UCAR-T and innovations like FastCAR. But what about the second hurdle? Efficacy, durability. If the therapy is cheap and fast, but the cancer comes back in 6 months, we haven't won.
Exactly. And this brings us to the rise of multi-targeting and TANCARs. The single biggest reason for relapse in traditional CAR-T is something called Antigen Escape.
Can you explain Antigen Escape for us?
Sure. Picture the cancer cell as a target painted with a specific logo, let's say CD19. The CAR-T cell is designed to hunt only that logo.
Okay.
Antigen escape is when the cancer cell evolves under that pressure and just stops expressing the logo. It sheds the CD19 marker, so it becomes invisible to the therapy. The cancer is still there, but the CAR-T cell flies right past it.
And the patient relapses. So the elegant solution is the dual targeting system. Instead of one target, you give the CAR-T cell two.
Precisely. We call these TANCARs Tandem CARs. They fuse two separate target-finding elements, called scFEs, into one single protein. So think of it like a specialized drone. A traditional CAR-T drone has one antenna for CD19.
Right.
A TANCAR drone has two antennas, maybe one for CD19 and one for CD22, fused onto the same frame, sharing one central control panel.
That single control panel, sharing one intracellular signaling domain, that sounds like a really critical design choice. Why fuse them instead of just giving the patient two different types of CRT cells?
Well, that fusion gives you two big advantages. First, the bivalency creates a synergistic, highly potent response. It finds the tumor better, it reacts more violently. And second, and this is the potential safety genius of it, some designs require the simultaneous engagement of both antigens for full activation.
So if the cell only finds CD19, it's not fully triggered, it reduces the risk of firing on a healthy cell that might only weakly express one of the targets. It's like a dual lock and key mechanism.
That's exactly right, an engineered safety margin. And we have a phenomenal case study here in multiple myeloma with Gracell's GC0112F product. It targets both BCMA and CD19.
And this was tested in a very tough group. Newly diagnosed, high-risk, multiple myeloma patients. What did the data show?
The safety profile was remarkable. Only 27% of patients had mild to moderate cytokine release syndrome grade 1 or 2. And critically, the sources reported zero neurotoxicity.
Zero. That's incredible. We often hear about much higher CRS rates, sometimes over 70%. Why was the toxicity so low here?
The data strongly suggests it's linked to the study design. These were newly diagnosed patients who got standard induction therapy before the SAR-T infusion.
So the initial tumor burden was already knocked down.
Exactly. Less tumor mass means a less explosive CAR-T expansion, so fewer inflammatory cytokines are released. It's a key lesson in managing the tumor load pre-infusion to maximize safety.
And the efficacy data, combined with that safety profile, that's what makes this so revolutionary.
It is truly stunning. They got a 100% overall response rate, and every single one of the 19 evaluable patients achieved a stringent complete response SCR and was minimal residual disease negative.
100% SCR in a high-risk population. That's an almost unheard of milestone.
It is, and we saw similar validation with another dual-targeting product, KQ2003. This one was in relapsed refractory patients.
And what did that show?
It was equally effective in the short term, 100% ORR. But its major contribution was the durability data. The 1-year relapse rate was only 12%.
Only 12%. What would you normally expect?
For traditional single-target CAR-T, you'd be looking at rates around 35%. So this really validates that multi-targeting is solving the antigen escape problem, which translates to better long-term survival.
And the commercial world clearly agrees.
The financial validation was immense. The tech behind that dual targeting approach drove a transaction that totaled $2.1 billion in development and commercialization rights.
$2.1 billion. That is not just excitement. That's global pharma putting a multi-billion-dollar stamp of approval on tankars.
It absolutely is.
So we have fast, we have cheap, and now we have durable and effective. But these are still lethal weapons. Let's pivot to the third critical area, standardizing safety. Because without that, these therapies can't move beyond a handful of elite cancer centers.
That's the reality. CAR-T toxicity, CRS, and CRS can be severe, even fatal. The need for specialized critical care and expert staff has been a major constraint, especially in rapidly expanding markets like China.
To tackle this, a major consensus was developed by multiple leading Chinese institutions, giving refined, standardized guidelines for managing these toxicities, specifically in B-cell non-Hodgkin lymphoma.
Yes, and they focused heavily on refining the classification of cytokine release syndrome. They introduced classification based not just on time of onset, acute, late, chronic, but also on location. Systemic versus a phenomenon they call local CRS or LCRS.
Local CRS, that sounds like a key distinction.
It is. LCRS is seen a lot in patients with a high tumor burden, and it shows up right where the lymphoma is most active. So instead of just a systemic fever, LCRS is redness, swelling, effusion right at the lesion site.
And if those lesions are in, say, the gut or near an airway, that localized inflammation becomes incredibly dangerous.
Incredibly. The swelling can lead to perforation or bleeding. This meant they needed very specific management protocols tailored to the location.
Can you give us an example? How do you manage LCRS differently than systemic CRS?
Well, take the GI tract. If you suspect gastrointestinal involvement, the consensus recommends gut purging and oral antibiotics before the SAR-T infusion even starts.
So it's proactive.
It's proactive. The logic is you prevent intestinal flora from migrating and fueling a much larger systemic CRS response once that localized tissue damage begins.
That's incredibly detailed, proactive care. What about a severe case, like in the neck area?
For severe LCRS in sensitive organs, like a huge neck mass swelling near the airway, they advise using anti-TNF-alpha agents. And critically, they recommend local interventions, like a tracheotomy, to manage the compression before it becomes a respiratory crisis.
That really highlights the stakes. Okay, moving to neurotoxicity, CRES. What's the standard path there?
The incidence is somewhere between 20 and 64%, and its severity is tied to high cytokine levels and tumor burden. The consensus confirms that steroids are the absolute cornerstone of treatment for CRES.
Right.
They're highly effective at calming that inflammatory storm in the brain.
And this leads to that critical distinction. Tocilizumab, the drug famous for treating systemic CRS, is not the go-to for CRS alone.
Exactly. The experts are explicit. Tocilizumab is not recommended for CRS unless the patient also has significant systemic CRS. For CRES, the focus is steroids. And they even recommend prophylaxis, giving high-risk patients levetiracetam to help prevent seizures.
Finally, we have to cover the long-term expected toxicities, starting with B-cell aplasia, the on-target, off-tumor effect.
This is an expected consequence. You kill the B-cell cancer, you kill the healthy B-cells too. This leads to hypogammaglobulinemia, so the patient can't fight certain infections.
So how do you manage that?
The guidelines standardize it. Prophylactic monthly intravenous immunoglobulin or IVIU until the B cells recover, or for at least 6 months. This is just essential, non-negotiable supportive care.
And what about general infections? The risk is huge.
Up to 70% incidence. Prophylaxis is paramount. Standard care includes protection against pneumocystis jirovecii, pneumonia, and zoster. But there's a crucial note for patients who are chronic HBV carriers. They need prophylactic entecavir for at least 6 months post-infusion to prevent the hepatitis B virus from reactivating.
That comprehensive framework is designed not just to treat toxicities after they happen, but to proactively manage and prevent them, which is the only way these therapies get adopted widely.
Exactly. These consensus guidelines represent a crucial, often unsung part of the innovation. You can invent the cure, but if you don't standardize the delivery and safety, it remains inaccessible.
This was a rapid yet really deep journey. Let's synthesize the knowledge nuggets you should take away.
We saw innovation tackling the 3 big CAR-T hurdles. First, UCAR-T is smashing the cost and time barrier. We saw massive single-batch production from Boya Bio and North Hang Bio. That fundamentally shifts the paradigm.
Second, CANCARs. We saw GC0112F getting those jaw-dropping 100% stringent complete response rates by neutralizing Antigen Escape. And that $2.1 billion deal confirms that the world believes in this strategy. And third, the safety protocols, specifically addressing local CRS and standardizing that long-term care like IVIG and HBV prophylaxis. That's essential for turning this into a real-world, scalable treatment.
So while multi-targeting is clearly key to improving efficacy, are there still limitations here?
Oh, absolutely. The field is still iterating. We haven't settled on the ideal time structure yet. Optimizing the design of these complex constructs is still an unsolved task. And, while the efficacy results are incredible, we have to remember much of this data is from small initial trials. We always need larger cohorts and longer follow-up to confirm these results.
So what does this all mean for the big picture? The next generation of cancer therapy isn't just about killing the tumor better, it's about making that lethal treatment repeatable, scalable, and safe enough for the mass market.
Which raises an important, provocative question for you to consider. We've seen these huge leaps in gene editing for UCART scale and massive success with multi-targeting for efficacy. What combination of those two revolutionary technologies, a universal, off-the-shelf product that is also dual or triple targeting, will finally tip the scales from this complex personalized medicine to a true off-the-shelf cure for a wider range of patients?